Hey guys, how are you getting on? It's been a little while since I've done one of these sessions, but with Easter. How's everyone? Hopefully you can see me. That'd be worrying. Or see at least a page and then maybe my face somewhere. Let's have a look. Oh yeah, no, I can see myself. This looks good. How are we doing? We're all right? Did anyone do anything remotely interesting over Easter whilst everyone's been locked up? No? Is anyone there? Ah, there you are. No? I came on the data free. I'm basically a wizard. That is what you need to know about me. No, I tried to get it pretty much dead on, and normally you have to go like a few seconds before to get it, but yeah. A lot of chocolate. Nice, nice. Well done. Chocolate's good. Yeah, I'm good. You wash your car for the first time in a year? Nice. Yeah, I should probably do that at some point, but I just can't be bothered. Hello. I see some of you are talking about dentistry versus medicine. I know there's like something that dentists can do. I think it's called like max fac or something, where you basically get a degree in dentistry as well as medicine. That's something which is a bit crazy. That's something if you are solely doing dentistry or medicine to sound better than everyone, that's what you've got to do. Also, like I swear it's like max fac, yeah, something like that. Hey Zara. What else do I see? Someone asked earlier if this is covering like all specs. Basically, I try and cover AQA, OCR, EdXL-A, and EdXL-B by taking exam questions from all four of those things. And then in terms of those people who are asking, like, what is this topic? It's like towards the start of year 12, there's like a little topic about water and ions. So I'm just sort of covering that sort of thing. What else were you guys saying? Someone said something fun, I'm sure. Maybe not. No. That's it. Maxillofacial surgery, yeah. Is there any experiences for medicine students available? I don't know. Fortunately, I've had a lot of experience with them. Oh, Alex, sorry you've had to deal with them, but they are pretty cool people I've had. I swear you get the example question every time now. Yeah, I do. I get the same questions every time. It's really fun. It makes my life really easy. Someone at some point will ask how long it's going to be as well. That is a standard question. What else? I think that's about it. How long? And I don't know, yeah. Maybe like, do I have slides? And then my man behind the keyboard will send the slides, something like that. Someone's been dancing. That sounds pretty fun. What have I been doing? I have been doing very little. Oh, I worked out the other day that, you're allowed out once for your exercise, and that's pretty much it. If I just run fairly far away, I can then have a nice leisurely walk back. That's been a nice little thing that I've worked out recently. That's been fun. Oh yeah, that's true. What we're doing next lesson, that's a good question too. Right, should we get cracking? Should we do some work? Let's do that. Hopefully you guys are feeling like you're still managing to get some like work done. I'm sure loads of you are feeling like really happy about not having to do anything, but for those of you that are here who are wanting to do stuff, let's do some stuff. Anyway, so, just so you know, and again, this is the same as pretty much always, at the end of this session, I'm going to have a coupon code for you of about 10%, which I have also purposefully not put the code in for just yet. So you're gonna have to wait for me to get there. So those of you that are looking at the PDF, you have to wait. Equally, what company does online work experience? That's really hard to find work experience for most degrees. So it seems like most people get it because they know someone, or you just hassle enough people that you eventually will get it. But anyway, yes, ions and water. So we're going to be looking at ions and water and going over some fairly straightforward things in my mind. So, that looks weird. That's resolution looking strange. Anyway, the photograph below shows apples on a tree. The mass of apples produced by an apple tree depends on the type of fertilizer used. Fertilizers provide inorganic ions required by the plants. Plants require the following inorganic ions to make the amino acids required for growth. So out of those, what do you need for amino acids? Hey, Masimi. Okay, what do you need for amino acids? So very few of you are saying calcium. Some of you are saying nitrates, some phosphates. Don't think anyone said sulfates. think about what an amino acid looks like is what I would say for this. So you've got a central carbon. You got your carboxylic group over here. You got your R group. You got hydrogen. And then what everyone should know is you have nitrogen. Okay, so that is what an amino acid looks like. So plants therefore require nitrates in order to actually make any amino acids. Now, some of you might be saying that there is one amino acid that actually does need sulfur in it, which is true, but generally for most amino acids, you don't need it. You just need nitrates. Okay, so nitrates is correct for this. So you need the amino group, you need a carboxylic group or the carboxyl group, and together you can use them to make amino acids. Okay, plants require magnesium ions as a component of what? Which of these things is used in, sorry, which of these things has magnesium in? Who knows? So cellulose, chlorophyll, phytochrome, protein. You guys know this. Nice, yeah. So magnesium is found in chlorophyll. There is a bit of magnesium in there which helps with the process. Definitely not cellulose, definitely not phytochrome, definitely not proteins. Well done. Easy, these are going to get harder. Okay, right, this one is a little harder and a little smaller, so sorry, this is a bit small. So the graph below shows the effect of different nitrate fertilizers on the mean mass of apples produced by Golden Delicious, nice, and Red Delicious apple trees. Using the information in the graph, describe the effects of the fertilizers on the mean mass of Golden Delicious apples produced. So using information in the graph, describe the effects of fertilizers on the mean mass of Golden Delicious. So that one. What is phytochrome? Phytochrome is just another sort of pigment, or is that the right word? Yeah, it's sort of a pigment which is involved in quite a few responses in plants. So it basically detects red light or far red and red light, and it uses it to essentially do certain things. So phytochrome is involved in germination. So when a plant sees red light, it will cause it to start to germinate, which is quite cool. So calcium nitrate produces Golden Delicious, I've missed it. Golden Delicious apples with a greater mean mass than ammonium nitrate. Okay, cool. So you guys are noticing that over here, ammonium nitrate causes a mean mass of 24, whereas calcium nitrate causes a mean mass of, what's that, 34. Okay. There is an overlap in standard deviation, so no significant difference when using calcium nitrate. Okay. I'm intrigued by that answer. The effect of fertilizer on the plants are able to photosynthesize more effectively. Calcium nitrate is more effective than ammonium nitrate. What time do the lessons start? Because I'm always late. Anissa, 3 o'clock, I believe. I think everyone is doing theirs at 3 o'clock, so me and Georgia. Okay, right. We need to say, we need to use some data or something, right? So we could say, because we're only looking at Golden Delicious, right? So I think one thing that a lot of people would make a mistake with this question on is you are not comparing Golden Delicious, against Red Delicious. So I think a lot of people would say, oh, calcium nitrate is at 34, whereas whatever that is, Golden Delicious is at whatever that is, 38-ish. So I think lots of people would start comparing them, and that's definitely not what you need to do. So I am just comparing the effect of calcium nitrate on my Golden Delicious and ammonium nitrate on my Golden Delicious. So I think you could say something like Golden Delicious. I really want an apple now. Golden Delicious apples grow. And then what numbers did we just have? So we had 24 there, and we had 34 there. Grow 10 kilograms more per tree. More with calcium nitrate opposed to ammonium. And then I want something about significance. So what can you guys tell me about the significance when we compare these two? Is there a significant difference, or is there not a significant difference? This lesson's just started, Anissa. So there is an overlap, or there isn't an overlap. Calcium nitrate shows a greater standard deviation, therefore they're less consistent. Yeah, but that's not the most important thing. Right, good. Because I am comparing ammonium nitrate, sorry, I'm comparing Golden Delicious with Golden Delicious. I don't care about these standard deviations and if they overlap. The ones I care about overlapping, the ones I'm going to change the color of so you can see it properly. The ones I care about comparing are those error bars and those error bars. Okay, so because this one here is not overlapping with this one here, it means there is a significant difference. So we could say growth is significantly greater with calcium nitrate, definitely not an M, as error bars do not overlap. Okay, so yeah, what you are looking for with questions like this, and I feel like a lot of people don't really realize this, is if your error bars are overlapping, so these guys, if you don't know what I mean, these two here, they overlap, right? They cover the same bit of distance. That means they're not significantly different. If they are not overlapping, in the sense of if you follow this one along here and along there, it doesn't overlap at any point with this one. That means that they are significantly different. Okay, so that's how you can tell if something is significantly different. You can use statistical tests and you can use error bars. Okay, so the significant, yeah, aren't they deviation bars? Does it actually say? Yeah, maybe I'm just assuming these are standard error bars. They could be standard deviation bars, but the mark schemes, the mark scheme I've got in front of me says that we are using the error bars to judge if there is a significant difference. Okay, so the Golden Delicious grow significantly better with calcium nitrate than with ammonium nitrate by about 10 kilograms per tree. Okay, the diagram below shows 4 molecules, P, Q, R, and S, found in living organisms. Draw a diagram to show the molecules provided when 2 molecules of R join together during a condensation reaction. I know you can't really do this too well. What's the important thing that I need to have drawn? So I'm going to have some water, okay? So let's draw, in fact, I'm going to probably do it up here because I don't have a lot of room. And I'd say that every single one of you should be able to draw this molecule, right? If anyone can't draw this, you need to draw it enough times until you can. I don't know why people decide they don't want to learn the structure of carbohydrates, but lots of people do this, and it's a bit irritating. CH2OH. Okay, and then I'm going to draw another one. Carbon, carbon, oxygen, carbon, 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 hydrogen, OH, CH2OH, H, OH. Okay, cool. So here is my here are my 2 glucose molecules. I am going to join these 2. So draw a diagram to show the molecules produced. So I'm going to take off this water, H2O, get rid of that, and then I'm going to have a bond forming between the C, the O, and the C. So let's put the O nice and central, and the C there. So you are looking for this sort of COC in your glycosidic bond. So if that is 1, which is going to get you a couple of marks. So let's have a look. Yeah, so you're going to get 2 marks for drawing this correctly and this correctly, and you got the correct bond. And then you're going to have another mark for saying the other thing that you make, which is H2O. Okay, so in terms of the carbons that you need to join, this is always carbon 1, 2, 3, 4, 5, 6. So carbon 1 is going to be joined to carbon 4. So 1,4 glycosidic bond, you got this O in the middle. How do you know the question is asking about glucose? Because it says 2 molecules of R, so R is glucose. What's that molecule called? It's called maltose. Is this EdXL? Yes, this is everyone. Everyone needs to know this. Like, there's no way that any A level would ever not let you know about glucose. Yeah, it is alpha glucose as well because the OH is at the bottom. Right, hopefully you guys are all cool with that. So hopefully this is also showing you that water's a bit of an interesting molecule because you make it. Okay, so the diagram below shows 4 molecules, P, Q, R, and S, found in living organisms. One of the products of the hydrolysis of molecule S is going to be what? A, triglyceride, B, an amino acid, C, glycerol, or D, water. Lots of you saying glycerol. So yeah, what are we going to have when we break this down? So when you hydrolyze it, you guys are right that it's going to be glycerol as one. What could something else be? Fatty acids. What else could you have with this as well? There's another term that gets sort of thrown about a bit. So glycerol is one, fatty acids are one. There's another molecule you can make as well. So fatty acids, glycerol, and I'll be surprised if you guys have heard of this. Yeah, monoglycerides. So sometimes when you break or hydrolyze this molecule, you can make something called a monoglyceride too, which is basically where you have 2 of the fatty acids have gone, but 1 of them is still attached. I was seeing if you guys would say water, because this is the water session. I wanted to see if you guys would say this. But hydrolysis is when you sort of use that water. So my water would have been, say, here, and I'm going to put one of the hydrogens with the OH on one side. So maybe I'm going to have a hydrogen here, and then this carbon is going to have the other OH attached. Okay, so you end up breaking this bond here. Carbon is going to have the OH attached, and this O is going to have a H attached. Okay, so yeah, be careful with that. Why is that? I just wanted to see if you guys would say it. I think it's I don't know. People get really confused with condensation and hydrolysis. Right, but hydrolysis is breaking water down, whereas condensation is making it. Why would that ester bond not break? Yeah, the ester bond. Oh, why does the last one not break? I guess not enough water could do it. I don't know. Likelihood of water being in the right place at the right time. It just sometimes happens. Okay, right. Next question. This is very water-based. So explain how the dipolar nature of water is essential for living organisms. No worries, Anand. So why is the dipolar nature of water important for living organisms? Whoops. What does dipolar even mean? And yes, Miss Emi, I do. So it allows hydrogen bonds to form. This is very true. This allows for cohesion to happen. Yep, this is true. It allows it to dissolve things. It allows it to be a solvent. Yes, agreed. It allows surface tension to be a thing. Metabolism, I don't think I'd say. I don't think this is the reason why it's a good metabolic compound. It's more to do with the other things too. So the dipolar nature, right? We've got my oxygen, OH, H. Oxygen is electronegative. Hydrogen is electropositive. And it means that essentially it can stick to other molecules, right? There's a slight charge difference, which means that it can sort of have this bond with other molecules, right? So there you go. There is bond. This is really important because it means hydrogen bonds can form, which all of you said, which I'd expect all of you to say. And this is important for one of several reasons. So you could say cohesion in the xylem. You could say solvent, I don't know, for transport of something. So maybe in the transport of enzymes, like globular enzymes or something. You could have mentioned specific heat capacity. That is why it has specific heat capacity. Which we could say is a useful buffer. Or we could say it is useful to maintain sort of environments for like animals to live in or something. You could also mention latent heat of vaporization too. Yes, Bethan. So there's only 2 marks. So to maintain body temperature, yes, but you'd need to say that that's because it has a higher specific heat capacity. Dipolar, it's got two of them. So positive there and negative there, and positive there and negative there. That's why it's dipolar. Expands when it freezes. That's not really very useful in biology. You are right that is a little fact about water, but it's not that important. Why is there a high latent heat of vaporization, or why is that important? The latent heat of vaporization is important in terms of us controlling our temperature. So if a lot of energy is required to make water evaporate, it means that when you sweat, it helps you to cool down. Adhesion, yes. So what does the Mark scheme say? The Mark scheme says it forms hydrogen bonds. This is useful because it's a solvent, allowing dissolving to happen. Cohesion/adhesion for liquid flow. Specific heat capacity as a buffer, and latent heat of vaporization causing a stable medium and for thermoregulation. What about the max density at 4 degrees Celsius? That has not so much to do with this right now. Okay, so give 3 roles of water in plants other than pollen tube growth. So it can help a pollen tube to grow. What other roles does water have that you guys are aware of? Solvent for transport, I mean that because it can dissolve things, it means that those things that it has dissolved can then move about. So glucose can dissolve into water, and therefore glucose can be moved about. If you had glucose as a solid in your body, you wouldn't be able to move it about very well. Okay, this is good. So lots of you saying metabolites. I think that is very, very good. I'd say that is one of the most important reasons that water exists and why we've used it so much as, well, basically why life exists, because water acts as a metabolite. So there are different ways to say this. You can say it is reactive. You can say that it allows hydrolysis to happen. But essentially, water is important because it allows pretty much every chemical reaction in your body to happen. So that is one important role of water in the body. Turgidity is important. So turgidity, we're talking about how water can have this role where it can move into, say, a vacuole of a plant, and it provides some form of structure because plants don't have bones. So that is something we could talk about. Yes. Respiration. No, I wouldn't say respiration is particularly important. Respiration is when you make water anyway, isn't it? You could say the opposite, which is photosynthesis. So it's definitely useful in photosynthesis, particularly in photolysis, which is where you replace hydrogen ions. You could have said transport, you could have said thermoregulation, and you could have said movement of sap. That's a bit vague, but yes. What is a metabolite? Something that can help metabolism, basically. Transpiration. So 3 other roles of water. The point of transpiration, though, is to sort of move water to different places, isn't it? So I wouldn't say transpiration is a role of water. I'd say that's what happens to water. But the actual physical role that water has is it can break things down, it can give structure, and it can carry out photosynthesis. Transpiration is just a movement of water. Why is being turgid a benefit? Because if you have enough water in your plant cell, it provides a structure, it provides support. Honestly, Zara, the Mark scheme is very, very basic. Hydrolysis, photosynthesis, sap transport, thermoregulation, structural support, and turgor. That's all it says. I wouldn't say that's the role of water, though. That's like a property of water that it will cohere together. I wouldn't say that's a role of water. It's just something that water can do. If you spoke about transport in terms of translocation, I think you could get a mark. So water is used to dissolve things, to move them, so such as sucrose, right? I don't think you get a mark otherwise. It's solvent, it's fine. Okay, so hydrolysis of ATP. This is a really good question. I saw this question earlier and I thought this would be a good one for you guys. So hydrolysis of ATP is catalyzed by the enzyme ATP hydrolase. A student investigated the effect of ATP concentration on the activity of ATP hydrolase. She used shortening of strips of muscle tissue caused by contraction as evidence that ATP had been hydrolyzed. She took 4 slides, A, B, C, and D, and added strips of muscle tissue of the same length to each slide. She then added the same volume of ATP of different concentrations to the slides and left each one for 5 minutes. She then recorded the final length of each strip of muscle tissue. So look, here are the different concentrations. This is how long the muscle is after she has put the ATP hydrolase on it. Okay, so you can see that essentially, as she is putting more ATP hydrolase on, more ATP on, she's getting more contraction. Okay. The hydrolysis of 1 decimeter cubed of 1 mole per decimeter cubed solution of ATP releases 30,500 joules of energy. 60% of the energy released during hydrolysis is used for heat. The rest is used for contraction. The student used 0.05 centimeters cubed of slide D. Calculate the energy available from ATP for contraction of the muscle in the slide. Right. Those of you who want to have a go at doing this, see what number you come up with. Otherwise, in fact, I'll give you like a minute to have a go at working some stuff out. I'll just read some of your answers. Adhesion is when water molecules basically will stick to other things, such as the xylem vessel itself. Cohesion is when water sticks to itself. Okay, good. So someone has got a good start to this. So if 60% of the energy released during hydrolysis is released as heat, then the first thing you need to do is find out what 40% of this is. That's a good place to start. Have a go going from there. So go on, yeah, I've done the first bit for you. Or someone has done it for you. So I've actually only got 1,000 or 12,200. Okay, instead of having 1 decimeter, I've actually got 0.05 centimeters. And instead of 1 mole, I've actually got 8 times 10 to the minus 6 mole per decimeter. So what am I going to do with this number? This is how I would do it. Yeah, so I need to do some converting, yeah? So if I know 1 decimeter of 1 mole per decimeter is 12,200 joules, then I want to firstly, well, it doesn't actually matter which one I change first, but I'm going to change my volume first. So I'm going to change decimeters, or 1 decimeter into 0.05 centimeters. So because I'm clever and I know this already, I know that 1 decimeter is the same as 1,000 centimeters cubed. So let's say that 1,000 centimeters cubed is the same as 1 decimeter, because it is, right? I know that because a decimeter is like 10, isn't it? So 10 times 10 times 10 is 1,000. Anyway, so if I'm going to convert that into 0.05, what number do I need to divide 1,000 by to turn it into 0.05? What number will I therefore need to divide 12,200 by? So what have how many times bigger is 1,000 than 0.05? I like it in my mark scheme for this. I've just said work it out. It's really helpful. Okay, cool. So 1,000 divided by 0.05 is 20,000. Okay, so if I've divided this by 20,000, Let's change the color to make it more obvious. So I've divided by 20,000. I need to do the same on this side. So divided by 20,000. Okay, so 12,200 divided by answer equals 0.61. Okay, so if I had only 0.05 centimeters cubed of 1 mole per decimeter cubed, I would have 0.61 joules. And then that one's already been converted, so I don't need to worry about it. I'm now turning 1 mole per decimeter cubed into 8 times 10 to the minus 6. So basically, how many times smaller is 8 times 10 to the minus 6 than 1? Okay, what do I need to divide 1 by to turn it into 8 times 10 to the minus 6? And therefore, what do I need to turn 0.61? What do I need to divide that by? Nope. Not 100,000. Why do I use 8 times 10 to the minus 6? Because I'm using D, so slide D, and this is the concentration of 8 times 10 to the minus 6. Good, 200, oh no, not 250. Yeah, I've got 125,000, right? So 0.05, not 0.05, 1 divided by 8 times 10 to the minus 6 is 125,000. So if I'm making it 125,000 times smaller, I need to do the same to this number. So I need to divide that by 125,000. Okay, so 0.61 divided by answer gives me 4.88 times 10 to the minus 6. Oh, you guys are being nice to super me. Yeah, so guys, that's how you work out the answer to this question then. So I would basically break it down into, firstly, it tells me that 60% is used as heat. So I work out 40% first, nice and straightforward. I know that 1 decimeter of 1 mole gives this many joules. So if I turn it into a smaller volume, I can work it out into that number of joules. And then if I'm lowering my concentration, do the same thing again, you end up getting this number down here. Yeah, it's only a bit of dividing. It's not that bad, this question. You guys should know that 1 decimeter equals 1,000 centimeters, or centimeters cubed. Okay, if you learn anything from today, learn that fact forever, or until you finish. Okay, the concentration bit here, that was fairly straightforward too, but that is your final answer. How do you get better at these maths questions? I also do, Kevin. You get better at these maths questions by just doing lots of them. So if you're not doing exam questions as part of your revision, you really ought to. So the more of these questions you see, the better you'll get at them. But 10% is going to be maths of your exam. 12,200, because they said earlier that 60% was used for heat. 60% of energy is used as heat. The rest is muscle contraction. Okay. I told you to have that question. Water and inorganic ions have important biological functions within cells. Give 2 properties of water that are important in the cytoplasm, specifically the cytoplasm of cells. For each property, explain the importance. So why is water important in the cytoplasm? Okay, so yeah, it is a metabolite. Why is that useful? So it is a metabolite, or you could say it is reactive, one of those 2 words. Why is it important that water can be reactive? What does water do for being reactive? What reactions is it actually doing? There you go. Someone said it. It's used, so it's reactive. It's a metabolite. This is important, so it can carry out hydrolysis reactions. Right, that's why water is needed for hydrolysis reactions. It does give a mark for condensation, but I don't agree with that. I think it should just be hydrolysis. So it can carry out hydrolysis reactions. Cool, that is one thing. Okay, in terms of transporting molecules, it's a bit important in the cytoplasm, but not necessarily the most important bit of it. I think you need to say something else. So what's another property? It is polar. Okay, property 2, it is polar. Why is that important in cells or in the cytoplasm? Water is used for other reactions, yeah, but it's hydrolysis reactions which are the main ones that it's doing. But photosynthesis being another one it could do. So it is polar, therefore it can dissolve ionic compounds, lovely. It can act as a solvent, which means it can dissolve things, or a universal solvent, helping to transport or something. Okay, something like that would be good. To dissolve substances, yeah. I think I'd want to say why it's important that it dissolves things. So it's important that it dissolves things to help to move them about, right? Transport materials in and out by osmosis. No, because it's only really moved, the water's only the thing that moves by osmosis. It's not really the other things dissolved within water that are being moved by osmosis, I wouldn't say. Okay, let's have a look at another one. We've got 3 more questions, I believe. So other than sodium, name one inorganic ion and give one example of its biological importance. There are numerous correct answers here. So name an ion and then tell me its importance. Okay, don't just tell me, tell me their importance too. Tell me their importance, don't just tell me the name of them. Cool. So yeah, Fe2 plus, hemoglobin, O2 transport. That'd be a good answer. What else? Hydrogen. Hydrogen is useful for co-transport. Definitely. That's a good answer. Sodium potassium pump. That's a protein. That's not an ion. Magnesium for chlorophyll. Yes. Calcium for calcium pectate. Tell me more. I don't know much about that. Calcium ions for muscle contraction. Definitely. K plus to maintain electrochemical gradients. K plus for NAK pumps. I wouldn't say that. What is the K and the Na used for in terms of neurons, if we're going to talk about that? Or what is it used for elsewhere? Phosphate. Good. That's a good one. I like that. PO4 3 minus used for ATP synthesis. That'd be good. Hydrogen ions for the electron transport chain. I'd give that. Chloride for chloride shift. I'd give that. Hydrogen for chemiosmosis. I'd give that. Hydrogen for ATP synthesis. I'd give that. Calcium formation of the middle lamella. Yeah, I'd give that. PO4 3 minus phosphate for the use of nucleic acid synthesis. Yes. Calcium for neurotransmission. I wouldn't give that. I'd say calcium. Well, if you said synaptic transmission, I'd give that. And if you said calcium for use of a neuromuscular junction, maybe I'd give that. Hydrogen for maintaining a pH, yeah, feasibly. Yeah, there's lots of right answers. I would say if you get a question like this, go for one of the most basic ones that you can think of. I would just go for that one naturally, right? Equally, something like that for I didn't even mean to do that. So PO4 3 minus phosphate for ATP synthesis, something like that. Just go for simple ones. You want to make sure, because it tends to be the case that the people who mark exam papers, they tend to have a degree, but you want to go for the most obvious thing for a question, in my mind. Okay, cysteine and methionine are amino acids. Chloride shift is something that some of you need to know about, but not all of you. It's basically a way to balance out a charge difference in red blood cells. Don't worry too much. Yes, I would say that knowing that it's Fe2+, it can also be 3+, depending on if it's oxidized or reduced. But 2+ is the one that carries oxygen about. Do you have to know the chemical formula, or can you write it in words? It says here, it just says name of ion, so no, you're fine to write it out in words. That's fine. Yeah, this is all needed for AQA. Oh, chloride shift, no, not needed for AQA. Okay, state and explain the property of water that can help it to buffer changes in temperature. So what property of water allows it to buffer changes? Hydrogen bonds is not a property. Dipolar, not really. So latent heat of vaporization kind of is to buffering changes in temperature. Okay, fine. Yeah, I would take either specific heat capacity. In fact, high specific heat capacity or high latent heat of vaporization, and then explain. So state and explain a property of water that can help it to buffer changes. Why does a specific heat capacity mean that it can buffer? Buffer sort of means it can keep it mostly the same, so it doesn't change too much. Good. So high specific heat capacity, explain in this property, lots of energy is required to heat up by 1 degree. Okay, that is important. That seems a bit weird. That's important because your body is mostly water, as I'm sure most of you know, like 60 or 70% or whatever it is. Most of your body is water, and if you went in like a sauna, right, where it can get to, I don't know, 100 degrees fairly easily, it would be pretty unideal if you just started to boil and evaporate and, like, I don't know, all of you started to just change state. That would be pretty not okay. So lots of energy is needed to heat water by 1 degree, and therefore that will buffer your temperature. It means that you don't basically die. High latent heats of vaporization. This is useful in terms of buffering our temperature because lots of energy, even more, is needed to make water evaporate. Okay, the question says, stating how that can help buffer changes in temperature. This is useful for us in terms of sweating because it means that sweating helps us to cool down, but it's also useful if you are like pond life or something and you live in water because it means that your habitat won't just evaporate when it gets hot. No, buffer I wouldn't really say, I wouldn't define as being control. Buffer is more to do with keeping it the same, so making sure it stays roughly the same. You put latent heat of vaporization for this in one of your mocks, you didn't give a mark. Ooh. But it's right though, isn't it? Like if you haven't, maybe you didn't explain it very well and you didn't talk about how it's useful in sweating or like in pond life zone, but it's definitely true, isn't it? Like by sweating, you're maintaining your temperature. Don't know. Okay, final question, I believe. Let's see what you've learned. So place a tick in the appropriate box next to the equation which represents the reaction catalyzed by ATP synthase. Whoops. I would guarantee, I did say pond life. I would guess most of you will get this wrong. Most people get this wrong, which I find really interesting. What does synthesis mean? Synthesis means making ATP. Bear that in mind. Making ATP. Right. That one is hydrolyzing ATP, so it's not that. That one is hydrolyzing ATP, so it's not that. So it's got to be between 3 or 4, right? What side does water go on? Does water get made? So condensation is what we're looking at. Is water made or is water used? Is water made or is water used? Condensation. Yeah, water is made. Well done, all of those of you that said that it is free. It is certainly free. It is definitely not for, right? Water is not used to make something in terms of making a bond. You need to take water out, don't you? If you remove water, then you can form your bond. So, I don't know, look, here's random molecule P, which has an OH attached to it. Here's other random molecule P, which has OH attached to it. If I want to make a bond between them, then I need to take water out, and then water is going to be added on at the end, isn't it? So I'm going to form pop, there you go, forming pop, and I'm making water, right? If you need to do that in an exam, just do it. It doesn't matter what the chemical is. COH, COH, right? I'm making a bond. I'm going to take water out. So it's got to be there at the end. It can't just vanish. So the water is going to be added at the end. And then I'm going to end up forming this molecule, which is now cock. Lovely. Okay, so you make water at the end, so it's therefore got to be this one. How did I know it was a condensation reaction? Because the enzyme's called synthase. Synthase means synthesis, doesn't it? No way that you're going to get away with forgetting that, right? Like, yes. So are you going to make more of these live videos? Yes, I am. Right. For those of you that have been thinking, this has been so informative, and yet I've enjoyed myself throughout this. Whatever could I do? Because I want to learn more A-level biology, but how do I do that? Well, I have an answer. For those of you that are interested in this, ta-da, I work for Snap Providers, who are there. I keep forgetting what shoulder they're on, one or the other. They are there, right? And we do lots of these sessions. So if you have a look at my web classes for this week, well, not even this week, for next week, look, got one on Monday, 2 on Tuesday, Wednesday, Thursday. They're web classes I do. So look, here are the topics that I'm doing, whole load of different things. As well as that, I do drop-in sessions. Ooh, if I can get my mouse to work, which is not, there you go. So I've got drop-in sessions where I've got something on Monday, Tuesday, Wednesday, and Thursday, 2 on Thursday, including a book club. Right? If you are interested in this, I would very much recommend that you guys sign up for Snap Provides. Not only do we have these web classes where I'm just teaching you guys, which is what I do, we also have like loads of basically videos all over our website where you can do a nice little quiz and you can see what you know. So here is something that's fairly to do with what we've just done. Whoops, and I just skipped a quiz, which was really done with me. And I've now gotten rid of it. There it is. So you do the quiz, right? And then, oh, I just skipped it again. I'm such a moron. Sorry about that. Maybe with Snap Providers, I'd get better. Right, here's a quiz, right? You go through the quiz, you have a go to the best of your ability and say, because you're clicking randomly like I currently am, say you get most of it wrong, right? At the end of the quiz, we have some really smart technology which will basically tell you what you do and do not know. So okay, nearly there. Should I do the last two properly? Well, they compare the length of those. RNA molecules are much shorter. Yes. Ha. There you go. So I apparently know nothing. Right, I got 1 out of 10 correct. I know nothing. When I click get started, look, it loads up all the videos that I clearly don't know anything about. So I don't know anything about these, and it's going to put the videos on. There'll be a man who will talk to me or a woman who will talk to me, and they will tell me things. So they'll go through this. There you go. They're talking through it. And then when you're done, there'll be another quiz that you can have a look at doing, and someone will go through this quiz with you. The quiz will be there. There'll be exam questions that someone will go through. And then you might be thinking, oh, well, I now know this really well. I wish there was a way that I could test myself. How could I do that? Well, there's an exam pack. And look, you could go on all of the questions and you could go and answer them. And they could be like, oh, I wonder if I'm right on this. I wonder if I know the segment of RNA shown in figure 1. And then, bam, there you go. It's a mononucleotide right there. So, yeah, this is Snap Provides. I am slightly joking here. But genuinely, it's a really, really good tool. So if you are at all interested and you like what you've seen today, then by all means sign up to our website. You can definitely get an account for free for a bit, where you'll basically get not everything, but you'll get some of the things. On top of this, if I haven't managed to sell this to you, because some of you are clearly have much higher what's the term? I don't know. Some of you, maybe you're not yet sold on SnapRevise. Maybe you don't think I'm quite good enough. That's fine. I'm cool with that. However, I do have more videos coming up in the next few days. So if you are not quite sold, then maybe it would be worth you coming to a chemistry lesson and seeing what that's like. Maybe it'd be worth you coming to a study in cells lesson, which I'm doing in the next few days. Maybe it'd be coming to, I don't know, something else, something about eukaryotic cell structure, right? If you guys set a reminder, you can come and watch me teach for absolutely free, but I will keep trying to sell SnapRevise to you because I think it is amazing. And look, you get a free trial anyway when you sign up. So for those of you that are struggling a bit and you are like, I don't know, not enjoying not having a teacher, then by all means come and check out our website and see what kind of things we offer. Apart from that, I've got like one or two more things to show you, don't I? Before I forget, not like that. There you go. Before I forget, look at all those questions done. Amazing. Yeah, so there are some videos coming up, so have a look at them. But otherwise, if you are interested, here is £10 off your first month. Whatever you choose, whether you have chosen to do like the best course that we have, which will have lessons, or if you choose one of the cheap ones, which just has exam questions, then use this and you'll get some money off. Okay, basically you're going to get your first month off pretty much for free anyway, I think, because I think you get like a little trial kind of thing. So you might as well use that and then have a go at using this and see if you want to stick around. I recommend that you should. Does anyone have any questions? Let's actually load up YouTube so I can have a look at your questions. Where are we at? Here you are. Oh, I may have lost you. There you are. Right, what were we saying? Oh no, that's definitely way behind. There you are. Yes, what 10? Thank you so much. Oh, no worries, guys. I'm honestly very happy to help. What else? So there are different packages for those asking how much is it. So different things will cost different prices, just so you know. Isaac, I'm glad I helped. Equally, Sammy, I'm glad I helped. I'm sure if you guys, I'm not even sure if you guys are still here. Next live biology stream will be next Monday. Does phosphate always help with strengthening bones and teeth in mammals? I'd say calcium does. Phosphate's more for like, I don't know, making DNA and stuff. Cheers, Ollie, lad. Nice. Right. In which case, guys, I think I'm going to end this here. So have a really lovely rest of your evening, and I will catch you at some point in the near future, I would imagine. See you later.